Hi, everyone. I'm grateful first to the organizers for inviting me and really looking forward to talking to you also after the talk. I hope we can have some nice discussions. Actually, let me, I'm a, do I have a pointer? I do have a pointer. So I'm a professor at the University of Maryland, but also I founded Biosim, some of you might know, and the Biosim's team is here. And also recently, I co-founded Entropy Sims. So I'm involved in both academia and the industry, and I'll talk about that a little bit as I go along. I know that many here have a computer science background, so I'll show you how computer science can help to understand better biology, chemistry, and with that, it can help us accelerate drug discovery and help to find cures. And this is going to be a high-level talk. I have many videos, so hopefully it will give you an idea of what it is. I don't want to bore you too much, so hopefully I'll succeed in that. So, but with that, I have to maybe introduce some terminology that we can do atomistic modeling because we are made from cells, and cells are made from molecules, and molecules are made from atoms. So we have to be able to model atoms and molecules. A new direction in science is to try to model the whole cell, and my group is one of the leaders in that area. And the third direction, some of you might have heard about AlphaFold and protein folding. So that's another direction I've been involved for a long time, and again, I'll talk about that. Hopefully I'll have time to talk about that at the end. So atomistic modeling, my personal efforts in that area, we've done lots of academic papers over the years, but in the last 3 years, my efforts in atomistic modeling were coming from a startup called Biosim. And Biosim started when I was doing a sabbatical in Armenia in 2020. And I worked with a group here called SmartGate. Some of you might know them. And we incubated, so they incubated Biosim, and I was the founder of Biosim based on my ideas of how we can put together some new technologies in computer science and machine learning and atomistic modeling to accelerate drug discovery. So bringing together a few things. At this point, this probably looks like a pretty picture, but hopefully I'll make some more detailed discussion of that. So at this point, I'll go into a little bit more technical discussion about why actually you need computer simulations in biology or in particular in drug discovery. So first of all, the computational challenge, like if you want to think about drug discovery as a computational problem, it's a hard problem. Why is it hard? If you say, how many organic molecules I can synthesize in a lab, theoretically, someone actually figured it out that it's roughly 10 to the 60th power. It's not exactly that, but it's like the number of atoms in the universe, like that kind of number. It's a crazy number. We cannot comprehend it. We'll never, ever synthesize that many molecules. So it's a huge space, and we have to search in that space. So that's why it's a hard computational problem. You have this enormous space, and you have this, and I think of drugs, medicines that are approved, think of, like cancers that are untreatable or Alzheimer's disease, like all these hard things. If you find, and I think many people think, including me, that all answers are there, that all the drugs that you could cure all human diseases, make us immortal, basically. They're all there. And those drugs are small things. Like if I knew what it is, I can draw on the napkin. And then you'll live forever. But so the whole question is, what is it that I should draw on the napkin? So it's a search problem. So the answer is there. The only question is how to find that answer. And in the last 100 years, we already found many answers. People used to die from lots of things. Now they don't. So we already made lots of progress. But as you know, we don't live forever. So there is work to be done. And I wanted to present it this way so that you could see that it's also a computer science problem in a way, that there is a huge search problem. And drug discovery needs to, so we have to bring computational thinking to be able to tackle it. So why is it difficult, just on a more practical side, to do drug discovery? This just gives you a rough idea of a preclinical drug discovery. Before humans get involved, before you try to get someone to take a pill and see whether they live or die. So you have to do it first with cells and then animals like mice and dogs. And so this is in the preclinical part without computers, meaning that these are biologists and chemists. There are maybe several dozen PhDs in the lab. And they think about some biology, like if maybe it's a particular cancer. They think about the protein. And then they come up, it's almost like chess grandmasters. Like chess grandmasters, like look at the chessboard, think, oh, like maybe this is a better move. And then someone says, no, like let's try another move. So they debate, they use a whiteboard to draw molecules. And let's say they come up with 200 molecules that they think is a good idea to try. It takes about 6 months to actually go in the lab. And organic chemists use these reagents, and I don't know if you probably have seen movies of that, to synthesize organic molecules. It might cost a few $100,000. It's actually probably more. It might cost $1 million to go through that. And then at the end, you see that most of it doesn't work. So you made 200 molecules, 190, those were just like, not that they don't work at all. And the remaining 10, they also don't work, but there is promise. Like you don't lose hope yet, or the management doesn't, because management can shut you down, right? Let's say you are a group at Pfizer, and you come and say, 190 doesn't work. We already spent $2 million, and it's 9 months instead of 4 that we promised. Then they could shut you down, and that's the end of this story. But let's say you convince them that there is 10, they don't work, but you think you can do something with it. Then you go to step 2, go to the board, the chemistry grandmasters come, and try to understand why those 190 didn't work. What was it in this 10 that they think? So they come up with another 200 based on those 10. And so this thing iterates. It takes 5. These are minimal numbers. Like it will go 5 years probably, this program, until you get either shut down or produce something. And it says 5 million, but it's more likely this is 50 million or 100 million to do this. And at the end, if you are lucky, you can get a drug. I just have to say that the possibility that at this point, the final point, 5 years, 50 million, you are lucky, it's about 5 to 10%. Meaning that at the end of this, with a 90% probability, you will fail. Okay? That's the state of art today. That's what with Pfizer, Merck, all these companies, this is the pain that we go through. And if you ask why medicine is so expensive, why do I have to pay $100 for the pill? It's because of this, that you are paying for huge amount of failed research. Okay? All right, so that's sort of one perspective. Second perspective is, how about we take a historic perspective? Like, we have computers, AI, like robots are going to walk among us, cars are driving themselves, like everything is getting amazing, right? Like, what's happening in drug discovery? Like, are we doing better? 1985, 2020. This is the efficiency of drug discovery. You can see that it fluctuates, and then there's some sort of linear fit. Doesn't look good, so things are getting worse, despite computers, algorithms, and all that helping, robotics. So something, something is not right, and some new approaches are needed. So clearly, or just humans are so complicated that we just should give up. But you also can see that this thing is going to extrapolate to 0 probably. Like it's becoming more and more expensive to produce a single drug over time. So, and you can see some statistics here about how much it costs and what are the failure rates and all that. Okay. All right. So the next. So coming back to science, or maybe even to computer science perspective, how we can formulate the problem. So to be able to solve a problem, you have to first formulate it. So I already told you about this huge search space. And what are we searching? What's the criterion? And for those of you that are doing machine learning, what's the loss function that you are supposed to differentiate, right? And here what it is that if you have a drug, let's say you came up with new molecule, and you have when I show this figures, there's always a disease. Let's say again, there's some cancer. An example today of a very hard cancer is a pancreatic cancer. 95% of people will die from that cancer within 5 years, even with the best modern treatment. So let's say you have an ambition to tackle that cancer. So what are you supposed to do? Let's say you come up with a drug that you think is going to inhibit a protein that drives that cancer. We actually know what protein drives that cancer. It's called KRAS. So let's say you think about either using computers or just thinking. You came up with a drug. And then there are two things that can happen. You are going to increase the concentration of that drug by giving someone a pill with more and more of that drug. So this horizontal axis is that you just give them more to eat. And that makes the concentration of the drug in your blood higher. So that's why this is the concentration. And then this shows the effect. Like, are you able to kill cancer cells? And let's say the answer is that after some point, if you take enough of that drug, let's say you start to kill them, and at some point you kill 100% of them. But almost all drugs are also toxic. You might kill a cancer cell, but also you might kill the liver, for example, and that's not going to work. So it turns out this is the red curve here, and it shows that after some time, that toxicity sets in. So this distance between toxicity curve and the good curve, the blue curve, this distance is called therapeutic index. And that's the optimization function you want to maximize, right? You want to make this distance large. If this distance is large, that means you are killing cancer cells and you are not killing the person. If that distance is small, then it's not going to work. I also, sorry, my voice is a little down. Yeah, as a point of reference, if you took a random molecule, then the order of these curves would be reversed, meaning that you first kill a person before you kill the cancer cells. So even this picture, it's very rare. It's very rare to find a molecule that will kill cancer before the person. And then after you found that case, then you want to maximize it and make it wide. So again, that's why it's a hard search problem, and we know what the optimization function is. Now, just a little bit of physical chemistry that, OK, how do we do this? We basically want to make, let's say if you want to kill that cancer cell, then your drug has to bind to that protein that drives cancer. In the case of pancreatic cancer, that would be KRAS. So, and this is how chemists measure binding. Like, your drug has to go and just stick to that protein. And the stronger it sticks, the better job it's going to do. So at the end, we assign some energy to that. So, and that sort of tells us how well that will do. And it turns out, now this is all experimental. At this point, you sort of say, how can computation help here? Is that, what if we could computationally screen drugs? What if in a computer, we could write a program that could simulate a molecule binding to a protein, calculate the binding energy computationally. And then, because if we could do that, then on an Amazon cloud, we could maybe screen millions of compounds quickly and get to the answer. Instead of five years, we can get to the answer in 3 months. So is it possible? So it turns out that when you do that, our simulation techniques have traditionally not been that good. So we have an error. When we make a prediction of how a molecule would bind to a drug or to a target, then we get an error, that energetic error, and and it turns out that that's not great. Like like the error, that error means there is noise in your prediction, and that makes it hard. So it helps to improve a little bit, but we need to do a better job. So basically, one thing we'd like to do is to take this error of a computer prediction and make it smaller. It turns out if you can do that, then you can accelerate drug discovery by 8-fold. So that's one obvious target. How can we do that? So again, and here I'm starting to go deeper into this computer simulation science. And one way to do that is called molecular dynamics. In molecular dynamics, let's say this is a target protein that you'd like to inhibit. And this target protein is embedded in a lipid membrane. So in an atomistic simulation, we need the protein, all protein atoms. Maybe there are thousands of them. Then we need all these membrane atoms. There are going to be maybe tens of thousands of them. And then we'll need water molecules that are going to be everywhere, and maybe hundreds of thousands of water molecules. So we are going to have tens of thousands to hundreds of thousands of atoms that are going to move around. And that's just atomistic representation. But then this is a picture. We want this to move. We want this to have dynamics. So for that, we have to describe all this chemistry in a computer. In a protein, this atom is connected to that atom, and they are at this distance, and then they vibrate, and they turn. Like all these physics and chemistry details need to be described. So if you haven't seen it yet, I don't expect you to sort of quickly say, oh, OK, I see what's going on. But I want to give you a picture also that I'm going to show you some movies that they are not actually Hollywood-generated movies. Like there's really detailed, deep physics and chemistry behind this. And our understanding of microscopic chemistry and physics lets us to run simulations. And then the funny thing is that we're actually going to use f of is equal ma to actually do the dynamics. So Newton's second law, I hope you remember your high school physics. We are going to use high performance computing to update velocities and positions to do movies or to do dynamics or binding for energies. And the thing that makes it hard, like why is it hard? Because this seems easy, f is equal ma. It's hard because this system has, let's say, 100,000 atoms. Potentially, it has 100,000 square interactions, right? Because things are, let's say, pairwise interacting. It's going to be less than that, but it could be, for example, I don't know, 50 million interactions. So you have to compute 50 million pairs of things. Again, computer science, right? You have a for loop of some sort, and you compute 50,000 times what's here. 50 million times, not 50,000. That's fine. Modern computers can do that. But then you have to iterate that billion times. So billion times 100 million, now like computers are going to have trouble. So that's why you have to have high performance computing. And the BioSim team knows how hard it is to write an efficient program to do that. This was abstract. I'd like you to see, actually, this is a computer molecular dynamics simulation. It shows how a drug enters into a binding pocket of a protein, not done by us, but just to give you the idea. That small molecule here that's sort of moving around, that's the drug. This bigger thing is a chunk. It's a chunk of a protein. And you can see everything is moving, everything is random. But this molecule is sort of, the bonds are rotating, the loops are sort of going back and forth. And what it needs to do for this to be a good drug, it needs to go in and find a place where it will dock and basically kill the protein, so that if this is a protein that causes cancer, for example, if this molecule is able to go in, like see it just went in, and it basically, it's almost like lock and key. So if it goes and finds the lock and the key goes in, then it basically can inhibit the protein. So this is a more now visual illustration of what we'd like to do. We'd like to be able to, A, simulate this process in some way, in a quantitative way. Ideally, we'd like a computer to tell us what molecule this should be, but maybe an organic chemist can tell us what molecule this is, and then we can simulate it. All right, this is BioSim's molecular dynamics. So again, as I said, the team is here. Over the last 2 years, we've been working on developing our own molecular dynamics from scratch. And the reason we wanted to do that, we want to accelerate it much faster than the best tool in the world that exists today, either commercial or academic. And at this point, we already are at 5x acceleration compared to anyone else. So, and these things, these simulations could cost millions of dollars. So if a simulation to find, let's say I'm screening thousands of drugs, and it's going to cost me $10 million, if I divide that by 5, now it costs me $2 million. So that factor of 5 makes a huge difference, both in terms of time and also in terms of money. And this is now a simulation coming from BioSim where you see a protein surrounded by water molecules, and you can see that how water molecules are moving around, how the protein parts are also moving. gives you an idea that these things are dynamic, they move around, they flex, and we have to have good computer simulation tools so that we can understand how they work, and also then we can understand how to design better drugs. We also we have a group at Biosim who works also on machine learning, and that's a different angle than molecular dynamics I was telling you about. There are now this, you know, that machine learning is now applied to everything, and you could say, why not apply that to chemistry and solve all problems and find all cures? And we are trying. And other people are trying. And it's not easy. Just pure machine learning, I don't think will really solve these problems. But if you take machine learning and combine it with this molecular dynamics and other things, I think altogether that could move the needle. So that's our philosophy at Biosim. So we don't believe you could just do pure AI, just throw data. You have no idea about chemistry and physics, throw data, just do some machine learning, and boom, you get all human diseases are cured. There are lots of companies that do that, and they get funded, actually, interestingly enough. We think you have to combine physics with machine learning. You cannot just do machine learning, at least at this, maybe in 20, 30 years you can. So we use machine learning to predict all sorts of quantities about, at the end, for example, you could imagine that, well, maybe we want to predict that toxicity, and if you have a drug molecule that your machine learning tells you that it's toxic, then don't synthesize it because you are going to just waste money. And another part of this drug discovery pipeline, and it's sort of multi-step, is that we have to take maybe millions of potential molecules and do what's called docking. So we first take millions of molecules, do docking, and try to quickly screen them. And then after we identify, let's say, 50 to 100 potentially good targets, then we use this more expensive simulations that I showed, like molecular dynamics. And at the end, we get to a few drugs that at the end, you take that to the experimental lab. So the idea would be that instead of the traditional way of doing drug discovery, we still have to go to the lab. There's no way that we could do this computationally only. But we want to accelerate it, meaning that instead of synthesizing 200 molecules right away, we want to study them computationally, and it will be quicker and cheaper to do it. And then instead of 200, maybe we only need to experimentally go and synthesize 25. And that will cut both time and resources to do the process, and it will accelerate drug discovery multiple folds, the preclinical drug discovery. This is first part of my talk, and I'll try to stick within 45 minutes, so I'll go faster from here. But I wanted to give you the background. So this is the team that's in Armenia working on this, and the SmartGate and Formic Ventures have been investors for us. I can tell you more if you want to talk about the company itself. All right, part 2, wholesale modeling. This one, I did this work over many years, both in my lab, but also I co-founded the company with Michael Antonov at Formic Ventures to do the following thing. We'd like to develop an open platform for computational modeling of all types, where we could have atomistic modeling, protein folding like AlphaFold, and also whole cell simulations, and make it open in a sense that other people that have these algorithms can come in and contribute. All right, so what is wholesale modeling? You can see this. Let me actually start again because this is fun. So this is called some people call this the most famous movie in biology ever. It was shot in 1950s. That's why it's so famous. Just imagine that. Now it wouldn't be that hard to shoot this. But basically, it's a human immune cell that's chasing a bacterium in blood. And you know, a single cell doesn't have, for those of you that are biologists, not biologists, a single cell doesn't have a nervous system, or a single cell inside has molecules, that's it. But it looks like an animal, right? It looks like it's thinking and trying to chase it and zigzag. And that's where I sort of, a famous physicist from 20th century said that about other areas of physics, that what I cannot create, I don't understand.
meaning that if I cannot create this cell, then I don't understand it, whatever biologists try to convince you otherwise. And because I'm a computational person, my sort of twist on that is what I cannot simulate, I don't understand. So if I cannot simulate this cell, what it was doing with this bacteria, then you just get these fairy tales in a biology textbook, but we really don't understand how cells work. So we'd like to have a model of the cell, and that's the current situation in science. We don't understand how cells work. This is another beautiful movie of two human immune cells interacting. And I hope you can see actually the pictures under this lighting. And the idea is that these things are very dynamic, and they come and they interact, and they chemically exchange information, they mechanically exchange information. And that's how our immune cells are fighting COVID and all these things. So we'd like to be able to simulate that. We cannot. 10 years ago, it would have been even preposterous to ask this question. This would have been science fiction. But this has become sort of, I think in 2020s, this will become possible. So maybe within 5 to 10 years, we'll be able to simulate what you see. And this was an experimental movie. And people have tried to do that, so I'll go fast. People have tried. Why is it hard to simulate? Because there are basically tens of thousands to millions of proteins in a cell, and they interact in very complicated ways, and you have to come up with this diagram of interactions and write all the programs and even get all these experimental details of who is interacting with whom and how. It takes billions and billions of dollars to get all the information you need to even put together a model. There is actually a group at Stanford that has something going on all the time on the web doing something like that. I have another friend, Zen Schulten at University of Illinois Urbana-Champaign. You can see it's almost like Minecraft. They used NVIDIA chips to, again, try to simulate a tiny bacterial cells, not human cells. No one can really simulate human cells. But try to simulate a tiny, tiny bacterial cells. And you can sort of see it here. It's almost like an electrical diagram. All these things, all these proteins are interacting with each other in a complicated pattern. And you have to put all that in correctly and have all the correct things and then have mathematics and then computer simulations to be able to really simulate, for example, how this bacteria grows and divides and all that. And my group is also leading in terms of development in this area. We came up with a software called Median, Mechanochemical Dynamics of Actin Networks. That can be downloaded and ran and all that. And I don't go too much, but it's based on, again, trying to model the whole cell as a molecular system. You can see that this is now the scale of a cell that we can do molecular dynamics of things. We need to have a membrane, so we came up with a membrane model. And also, many proteins, receptors, for example, signaling, they diffuse around on the membrane, so we have to be able to model that. This work came out last year on the cover of Journal of Physical Chemistry B, from my academic lab at University of Maryland, describing the breakthrough that we had in this direction. And this is basically what it looks like. This is a little bit smaller than a human cell. But it shows that it's a computer simulation, again, not a Hollywood movie, of things coming together. And you can see that you have a membrane, you have things inside, and the idea would be to simulate cells in this way. And what we already have scientific collaborations with biologists. This one is at a lab at NIH. This is a growing neural axon. So when a fetus develops, in this case, actually, it's a Drosophila embryo. When a Drosophila develops, then a neural axon, it goes in a wing. So you have a Drosophila has a wing. So and they put it under microscope. They can see one of the axons from a neuron. It goes like this, and then it makes a 90-degree turn, and it goes to a specific place. That's how all our neurons grow to get to our fingers and all that. We don't really understand how that works. So we are working using those models that I told you. We are trying to understand this at a molecular level, like what is it that makes this thing to turn 90 degrees and all these things. So we are doing that kind of model. Another collaboration I have with a colleague at University of Maryland, Arpita Upadhyaya, where they are doing experiments on T cells, which are, again, immune cells that when you have a flu or COVID, it's these cells that fight, the T cells, they fight infections. And we are working with them. You can actually see So we are working with them to understand how they work. And you can see now this is a computer simulation that we do that sort of tries to explain how T-cell activation, parts of T-cell activation work. And this is another movie that you can see sort of this again, the T-cell activation and our computer simulations of how you can see that some cells shrink, some cells don't. We just did a work, very detailed work showing that we can explain many, many processes there. And this last part that I'll go fast, and I think I'm going fast actually, is protein folding. I just want to give you a high, of course there are lots of details in any of these parts we can go deep, but I want to give you a high level overview of this kind of modeling. Another program that my group with a group of Peter Wolness at Rice University has developed over many years, this is now related, for those of you that know, related to AlphaFold, like how can we fold proteins and look at protein complexes. And the way we have done it is, and this is interesting because Peter Wolness, who is he was my postdoctoral advisor, he did this work in 1989, published in Science. And he said, is it possible to use a Hopfield network, which is a kind of a neural network, to model protein folding? And for those of you that know, that's basically what AlphaFold did in 2018. So, and already in this work in 1989, he had already some successful parts to it. So this whole field, for those of you that know, DeepMind didn't invent it. This thing has been going for decades with really amazing results. They actually took lots of that stuff from our papers and others' papers, and then they added superb engineering to it and got really nice results that now are rightfully acknowledged as being a breakthrough results. But it didn't come, those results didn't come in vacuum. They came hard scientific work of dozens of laboratories over the world over decades, and successful work. This is our paper that describes how that works. Aram Davtyan, who was, I know that he's in Yerevan, but he's a research scientist at Atomwise in California last 3 years. He was my graduate student, and he did this really cool work. And I'm just going to show you again some pictures that let me first explain what a picture is. You see, let's take this, that proteins have names. So this one is, let's say, it doesn't matter, biologically it's not important what it is, but let's say this protein has a name 1R69. People went and did X-ray crystallography in the lab to purify the protein, to crystal. You make crystals, you put it on the X-ray microscopes, and after some hard work, and it's not easy for many proteins, you cannot do that. For most proteins, you cannot do this. For this one, you can. You get a crystal structure that's drawn in this diagram here. You can see that there are two things, right? There are two structures on top of each other. One of them is the crystal structure, meaning this is the right answer given by nature. The second one, you can see that there is a superposition. There is another one that's put on top of it, is the one that we predicted using AwesomeMD. And so again, so this is, you have a computer prediction versus the ground truth, which is nature's result. And you can just visually see that this looks good, right? Like we had a really good prediction of how, in what way this protein will fold compared to the ground truth. We can look at many other proteins. So in some cases, if you see this superposition, for example, we get right. You see that the shape is more or less right, but there are like this helix, for example. It's slightly shifted. So there are these small mistakes, and there are techniques in some cases for the same protein to correct it. And then you see when you do that, then the computer prediction and the experiment come out to be very similar. This shows now multiple results. So this could be just one-way thing, but this shows a result where now we have multiple proteins. And you can again see that for some proteins we get excellent, and these are, just look at it, these are complicated shapes. And let me actually tell you how these simulations work. You just give me a protein sequence, I turn it into a line, think like a spaghetti. And then you do molecular dynamics, so this spaghetti moves and does crazy things. But then at the end, it folds into this shape. Unique shape. If you fold any other shape, it will be the wrong answer. And then you compare it with the crystal structure, with the ground truth, and you can see that, I mean, it's not perfect, but to be able to get that shape 20 years ago, again, that would have been considered science fiction. That it's like if someone said like try to do it by hand, like I give you a sequence, try to do this. It's very, very complicated. The same thing happens actually with, you might ask a question about protein complexes. So two proteins, like most biology works when two proteins come together, they interact. They again interact in very specific ways. One protein recognizes another protein and comes and docks this way, but not that way. And it's really important to get that right. Again, in these pictures, using AwesomeMD, we predicted what those interactions should be, that final three-dimensional shapes of two proteins come together. That's one color. Let's say, just for the argument, that's the blue color. The yellow color is the ground truth coming from experiments. Look at all these results, how our predictions compare with experiments. And this was published actually in 2012, way before AlphaFold, right? This just show you some movies that these kind of simulations you can use to see how proteins are not static objects, they move around. If you don't get that, you are going to miss most of biology. And AlphaFold cannot do this, even today. So that's why things like AwesomeMD need to be developed further, because they can do things that AlphaFold cannot do. And I'm almost done. More complicated things just that we can simulate with this is called, this will be the last slide. Anyway, my computer is not happy with something. But anyway, just to say that, there are movies here that are not playing for some reason. But this, for example, this is a way to model epigenetics. You know, we have genetics, we have epigenetics. And the most famous example of epigenetics is that if I think in World War II, when there was hunger, the kids that were born had epigenetic modification, and that predisposed them to diabetes because there was hunger, and the mothers who had these kids were under stress. That went 2 generations. So the way how that transmits is via epigenetic modifications. And these are kind of things that we can simulate too. Maybe I'll just leave with this picture. We published another really interesting paper with this epigenetics. And just maybe to tell you that when we computed computer simulation results, which are gray, with experimental measurements, we can see that we can get close agreement. So this is all hard work to do computer simulations. 10, 20 years ago, most of the work that I showed you today would have been insane. Like this is really new stuff, last 10, 20 years, that we're able to not just make pretty movies or just make conceptual statements, but really simulate biological things, compare it with experiment, and see that we get roughly the right answer, or that roughly the right three-dimensional shapes, or if it's a T cell attacking a bacteria or something, that we are starting to describe that also correctly. So, and we are still, in some sense, we still, I think, need at least another decade before this becomes real engineering. So, in a way, how would you, for example, nowadays design a computer chip or a car? You do it in a computer. You use this complicated engineering software to design car parts, engine, this, or if it's a computer chip, the 3D chip design. And you do most of the work, in some sense, in a computer. And only when you have a really good sense of that you have a good product in a computer, then you go and design and synthesize it and test it. And that accelerated chip design, that accelerated automobile industry, aviation industry, several-fold. It hasn't happened yet in biology and drug discovery. But I'm hoping that next few years will be the time when computer simulations will also accelerate drug discovery in the same way as for cars and chips and airplanes. So, thank you.
Okay, you can ask your questions. There are 2 microphones. Our volunteers will help you. So while you're going to approach microphone, I would like to ask, where are we as humanity in a discovery of like cure for cancer or something like that?
So I think on the positive note, I think lots of cancer cures, you know, cancer is basically 1,000 diseases. So for some specific cancers, cures were almost found or breakthroughs were found. So things that 20 years ago, if someone got that cancer, that would be terminal. Nowadays, it could be almost turned into chronic disease. You have to take some drug. But then there are cancers like pancreatic cancer that doesn't matter if you are rich or poor or wherever you live. If you get it, then you die, basically. So it's sort of the glass is half full, half empty. But there are thousands of diseases that we don't have cures, including cancer or Alzheimer's. You know, if someone gets Alzheimer's, there's no cure for it. And they just lose their memory, and lots of, unfortunately, older people get that.
Is there a prediction on like next 5 years?
No, I think so. But my prediction is that unless you do this, it's going to be hard to solve that problem. So you have to be able to do computational drug discovery. So there is computational drug discovery. It just doesn't work very well.
Yeah.
But if you had computational drug discovery that worked, then I think you could tackle Alzheimer's or pancreatic cancer. I think that would be easy, actually. So that's what I think needs to be done.
Thank you.
Hi, thank you for the talk. My name is Alexei. I worked at IPA, and now I work at Quantori with international pharmaceutical companies. And currently, you have already mentioned that you use the HPC for the simulations. Of course, they're needed because that's a complex structure to simulate. I wonder, how is the HPC organized at Biosim and maybe University of where you're working? Is it on-prem solution, or do you look into the clouds like Amazon or any other platform?
Yeah, at universities, we have this HPC solutions that are this multi-million-dollar or maybe $10 million clusters that we use to run some of these simulations. At Biosim, the idea is that we have some local small HPC to do code development, software development, and testing servers. But then the large-scale drug discovery would be done in the cloud. And when we do development, so one maybe difference is that in academia, you often might ask a question, can I parallelize this over 10,000 cores? Because you are going to try to simulate the whole virus. Often it has little scientific value because it's going to be still too short for any interesting biology anyway. But people who do it just because it's cool and you can do maybe 1 billion atoms and all that. In drug discovery, you don't need that. In drug discovery, you might need to do, let's say, 1,000 drugs. And they are, in some sense, perfectly parallel because you could have 16 core machines. And each of them, you do sort of a, let's say, 2-day simulation. And you need 1,000 of those. So you just throw them into the cloud that way. So I think the needs and the culture are a little bit different in terms with regard to HPC between at least drug discovery research in the industry versus some of these big simulations that people who do in academia. So that.
Thanks.
Thanks for an interesting talk. So you mentioned that.
Is it on?
Hello? Can you hear me? Can I ask?
I can repeat your question if you.
Yeah. You mentioned that Biosim MD runs about 5 times faster compared to other MD simulation softwares on CPUs. What about on GPUs? Because most simulations currently done on GPUs. And how does it performance compare to other, like specially designed chips like Anton?
Yeah, so these both are very good questions. With regard to GPUs, actually are on a small CPU, on a small CPU system that has 8 to 16 cores, we can already run it faster than a GPU, what people do. But we wrote our code in a way that is GPU, that basically, so that even though we developed it for a CPU, we also had GPU in mind all the time, so it should transfer very directly to GPU, and that's one of the projects this fall for Biosim to also implement it in a GPU. So what I'm saying is that for a CPU, we already have a solution that, in some sense, that's superior to actually what GPUs do, at least from the viewpoint of economics, that we can run it cheaper than people can run it on a GPU today. I think when we implement it on a GPU, our software, we have to see actually that whether that economics still works that way or whether the GPU for us makes things, because GPUs have become quite expensive lately. So that's that. And I have the same answer for Anton. Anton can run a lot faster than, let's say, a GPU simulation, but it also costs, I don't know, like 1,000 times more than a GPU. If you divide that, if you say, what's the price of 1 microsecond per dollar in Anton versus GPU, then that's why they don't sell Anton, right? Like, you cannot just go and buy services from D-Show so that you can run Anton simulation. They don't sell it to you, because if they sold it to you, you probably wouldn't have money to buy. So they invested $500 million of their own funds as basically just scientific research internally. But I don't think it's sort of easy to compare that to either CPU or GPU solutions that are large scale and that, for example, pharma companies can buy to do drug discovery. So at least in our hands, what we see is by far the most economic solution at the moment for us, and at least for us compared to other people, is the CPU solution, even compared to other people's GPUs or to Anton. When we do a GPU solution, then we'll see whether that changes. We are not actually sure. It could be that CPU solution is the answer.
I see, thanks.
Yes.
Hello there, thank you for your wonderful presentation. My name is Nadina, and I'm working in healthcare communications. My question is, you're working with very complex things, and I guess that the price of the mistake could be very high. So how are you dealing with the heaviness to making right decisions? Thank you.
Thank you, and that's the reason why the cost of finding a single drug is $2 billion in 15 years, right? If it wasn't that, then it would be really quick. But then you want to be careful that you don't harm people when you develop a new drug. So you want to do a human trial only when you are absolutely sure that it will work. Of course, when you go and do a real trial, you see that most of the time it doesn't. But at least at the best of your knowledge, and the way you do it is, let's say you test your drugs on cells. And you see that it's toxic for cells. Then you stop. You don't go further. You try something different. Let's say you found something different, and it's not toxic for cells. You have some hope. You put it in mice, and it's toxic for mice. Unexpected. You didn't know why. They die or they get liver damage. You stop. You go back. You think. You do a new design. Come back with a new drug maybe 2 years later that is not toxic in cells. It works. It kills cancer. And it's also not toxic in mice, and it kills cancer in mice. Then maybe you go to dogs. So that's why it's so expensive and long to make sure that at the end, let's say you have a drug, it was not toxic in mice, it was not toxic in dogs, it was killing the cancer in both. And at that point, you might then give it to pancreatic cancer patients. You know, many pancreatic cancer patients have a 2-month lifespan, meaning they get a diagnosis, they might die in 2 months. So at that point, when you test it in mice and dogs and it was safe, maybe then it's worth trying with pancreatic cancer patients, because in that case, for example, it might be worth the risk. And it also depends on the disease. Like with pancreatic cancer, you might be more aggressive because they only have 2 months. But then if it's some other disease that it's just some chronic disease, then you want to be even extra careful, right? Make sure that whatever disease you are curing, you are not causing harm. So that's exactly that question. That's why it costs so much and takes so long to develop a drug.
Hi, thanks for the very interesting lecture. So you showed us a graph where the efficiency of drug discovery was gradually declining. So what do you think is the reason for that, or several reasons? Is it because all the easy targets have already been discovered, or there are some other external factors not necessarily related to scientific activity?
Yeah, thank you for a very interesting question, because that is one of the questions that lots of people debate, because no one really knows the answer. You can just hypothesize. But people that study drug discovery research, they do have 4 or 5 hypotheses. One of them is what you said, that easy fruits have been taken, and now we have to deal with hard things like Alzheimer's. That's true. But there are other things too. It seems that actually in 60s and 70s, maybe people were approaching drug discovery in a smarter way. And then what happened, actually, the funny thing, what happened in 80s, 90s, and 2000s, people tried to automate it. So instead of having a grandmaster organic chemist that was like a genius, you say, well, I don't want to pay this, whatever, $300,000, it's too expensive. Let me buy a robotic kid that will, instead of this guy scratching his head or woman, right, thinking what to do, let me just try to automate it, have high throughput screening. Then I don't have to think hard. I'll just try lots of molecules, and I find the answer that way. there is an indication, and the whole industry actually had a transition to that model, spending hundreds of billions of dollars, thinking that this is a more superior modern model, robotics, high throughput screening and all that. But now people are starting to wonder, like, was that wrong to do that? Like, was it better to have really smart people just look at the blackboard and think instead of trying to do robotic high throughput screening? And the whole reason why that's the case is that the search space for organic molecules is 10 to the 60th. And if you are doing high throughput screening of 100,000 compounds, that's nothing in that space. So maybe it's better to have a really clever person to think in that space versus try to do robotics and high throughput, or maybe to combine. But now we are thinking that maybe if you do computer simulations or machine learning in a clever way, you can address that. But clearly, yeah, clearly something didn't work the way people thought it would. And I'd say, I think it's going to be, and also the third thing is regulatory thing, that regulation and safety have become much more strict last 50 years. So things you could get away in the 60s, when you do human clinical trials, you had to jump through a lot more hoops today. So probably if you take those three things, if you take easy targets were taken, the business sort of minds of pharmaceutical company probably took it in a completely wrong direction.
And bureaucracy.
And then and it's maybe good bureaucracy because, you know, like you don't want to kill people in clinical trials, right? So we are more cautious today as a society. So if you add these three things, then it makes drug discovery more and more difficult per drug.
Since you mentioned Alzheimer's, I have another question. Wouldn't it be easier to look for targets that are actually preventing the disease, like brain aging, for instance, targets that reduce brain inflammation or treat a leaky blood-brain barrier instead of treating Alzheimer's because that's much more complicated?
I think these are the hottest targets people are thinking about. But, you know, also to be able to work on it and then to see if it worked, it takes, because Alzheimer's takes 20, 30, 40 years to develop. So it's also really hard to, let's say you have some idea about leaky barrier or something. Maybe you can do a test on mice and it sort of works, but then how do you prove on a human that it's useful? So you might need to do a 20-year clinical trial. So that's, I think, that's the difficult question there, how to, yeah.
Maybe that's also a regulatory problem if these things were considered diseases, like brain inflammation was a disease itself and the leaky blood-brain barrier instead of Alzheimer's. Then you could run actual clinical trials with endpoints and measure if there is improvement.
Yeah, I agree, but I think people are starting to think about these things, and there are conferences that come together to address these questions. Yeah, I think it's a very hot area now.
Yeah, thank you so much.
Thank you.
Thank you.